Hi, my name is Liz Carlsen, and today I'm going to be talking to you about standardizing transformation efficiency and demonstrating FLG22 internalization. To start off with, plant transformation is a very useful technique. We use it from everything from basic research to crop improvement. However, traditional plant transformation techniques have two main issues. First off, they're qualitative, which means it is difficult to compare them across species and across samples. Number two, they are inconsistent. Things like magnification level, the gain, and the exposure can all change the quality of the image. In order to limit these two things, we are attempting to create a transformation efficiency protocol. To do this, first we needed a protein that plants do not normally express. We used luciferin, a firefly protein. Next, we needed a way to measure those proteins quantitatively. We are using reactive light units. Then we needed a way to get that protein expressed within plant cells. We are using the traditional agrobacterium-mediated transformation. Once we have these transformed plants, we take leaf discs and we find the total amount of protein using a BCA assay, and we find the amount of luciferin using a luciferase assay. The results of that are here. As you can see, they are lovely linear. More protein concentration means more luminescence, and then larger leaf diameters also mean more luminescence. Huzzah! Now that we have a standardized protocol for transformation to measure transformation efficiency, we can compare different types of transformation protocols. The traditional transformation protocol is agrobacterium-mediated transformation. This is when we have a bacteria with special DNA in it that we can use to infect cells. It inserts its tDNA, and then it causes expression of that said foreign DNA. We are also proposing a theoretical lipid nanoparticle-mediated transformation that should work in more species than agrobacterium does. For this, we have a lipid nanoparticle here, coated in FLG22 receptors, the brown squiggles, that are attached to FLS receptors and then cause endocytosis of said lipid nanoparticle and the DNA, theoretically leading to expression. In order to do this, though, the first thing we need to check is that FLG22 actually causes internalization of something it's attached to. In order to do this, we are attaching the FLG22 to a dye, a fluorescent dye, rhodamine, and we are soaking leaf discs in it. When we soak leaf discs in this FLG22 receptor, we see these lovely little pinpricks of light. These pinpricks of light suggest that they are endocytosed into an endosome and are actually inside the cells. When it is just the dye, no FLG22, it is just this basic background with no specific pinpricks of light. When we add just water, there's no fluorescence, which would make sense. And when we tried it in a different species, we got the same result of said pinpricks of light. Although this suggests internalization of the FLG22 and rhodamine, it does not confirm it. So we need to do further studies in that regard. However, if we do confirm internalization, we can conjugate the LMPs to the FLG22s and see if the LMPs get endocytosed as well, which would be a huge step forward in the creation of a lipid nanoparticle-mediated plant transformation. I would like to acknowledge the Cornell College of Biomedical Engineering as well as the Alabi Lab for their invaluable help on the project. Thank you very much.
So what species did you do this originally in, and do you think it could translate across species as well?
Yes, all of these photos are within Arabidopsis, and then when we did it in a second species, Benthaminiana, we saw the same results. Theoretically, lipid nanoparticle-mediated transformation should work across many different species since everyone has that same cell membrane. Originally, this is used in mammalian cells. Now we are trying to use it in plant cells. So hopefully it will work across a wide variety of plant species.
Can I ask one more question? Do you think this would be true for both like C3 and C4 species?
I believe so, because again, the cell membrane is roughly consistent across multiple spectrum of life. So hopefully that is the hope.
Thank you so much.